Hello everyone, welcome to the class. In today's lecture, we are going to talk about glycolysis. So let's start with the same. As we know, glycolysis is a Greek term which means the breakdown of glucose. Glyco means glucose and lysis means breakdown. So what completely glycolysis is? The breakdown of glucose to produce pyruvate or pyruvic acid along with ATP, because this is the part of respiration, and as a result of respiration we'll be getting energy. So this glycolysis, where we are breaking down this glucose, will be yielding firstly the pyruvate by the partial oxidation. Got it? This glycolysis is also termed as Emp pathway after the name of the scientist Emp, Merhoff and Parnas, as all these three were involved in the discovery of this particular process. That's why it is Emp pathway. This is the common pathway which is going to occur in both the types of respiration. Initially, the glucose is going to break down into pyruvate, and afterwards there are the different fate of pyruvate, that is depending upon the conditions within the cell, that O2 is available or it is not available. Got it? So common for both. The site where this glycolysis is going to take place. As we are talking about cellular respiration, so within the cell, the glycolysis occur in cytoplasm. Fine. And now the question is, in the case of plants, the plant prepare the food in the form of glucose, and if this is going to get oxidized, there will be no storage of the food. So what happened, the food which is transported to different sites of the plant, like from the leaf to the root, that is in the form of the sucrose. Sucrose is a disaccharide made up of two sugar units. And what are those sugar units? One is glucose and one is fructose. So the glucose which is going to get oxidized or which is going to undergo the glycolysis process, this is coming from this sucrose. Okay, so both these glucose and fructose can enter into this particular pathway. Now we are going to talk about the detailed 10-step process of glycolysis along with the enzymes involved. Now we are going to talk about the steps of glycolysis. Glycolysis is a 10-step process which is facilitated by enzymes. So let's start with the same. This glucose is a substrate which is going to get partially oxidized during the glycolysis to form the pyruvate. So the very first, this glucose is converted into glucose 6-phosphate. It means a phosphate group is added to this glucose. This is phosphorylation of glucose. And this phosphorylation is done by the enzyme called hexokinase. Okay? Enzymes which help in phosphorylation, they are kinase. So this one is hexokinase. And by the use of one ATP molecule, ATP is utilized, ADP is formed, and by using this ATP, this glucose molecule get phosphorylated. In the second step, now this glucose is converted into fructose 6-phosphate. So fructose is a isomer of glucose. It will become fructose 6-phosphate. Why this is done? This is done to increase the side chain number so that the modification can be done in the structure. Glucose is a 6-member ring only having one side chain. Fructose is 5-member ring. It is having 2 side chains. So if I draw the structure in this way, one side chain and this is having two side chain. So at two position the phosphate can be added in the fructose molecule. So that's why it is isomerized. And as it is an isomer and isomerization is occurring, this is facilitated by the enzyme called isomerase. Got it? Now, in the next step, this fructose is again get phosphorylated at that second side chain to make the molecule. Now will be forming fructose 1,6 bisphosphate. Bisphosphate means two phosphate groups are added over here. Okay, and this is again going to happen by the use of ATP. So two ATP molecules are used till now. ADP is formed. Got it? Fine. And this is facilitated by, because phosphorylation is occurring, so kinase. Which kinase is? Fructophosphokinase or phosphofructokinase. This is, let me write with the another ink. This is phosphofructokinase enzyme. Fine. In our next step, this splitting of this molecule is going to occur. Fructose 1,6 bisphosphate splits into two molecule of 3 carbon. Okay, what are those molecules? This splits into 3 phosphoglyceraldehyde. It is going to form 3 phosphoglyceraldehyde. Okay, or 3 glyceraldehyde phosphate. Another molecule which is formed by the splitting, that is dihydroxyacetone phosphate. Okay, this is also called DHAP. Both these molecules, glyceraldehyde phosphate and dihydroxyacetone phosphate, are 3 carbon containing molecule and they are interconvertible. It means dihydroxyacetone phosphate can be converted to form this, and this can converted into DHAP. Got it? So after this we'll be going to continue with the 2 molecule of this. Okay, either we can continue with this, either we can this, so we'll be taking 1. So when we're going to continue with the further steps, we'll be taking 2 molecules. So now this molecule, when the fructose 1,6 bisphosphate splits into 2 different molecules, one is DHAP and one is this 3 phosphoglyceraldehyde. As I have told you, we are going to continue with one out of these 2. So 3 phosphoglyceraldehyde now get oxidized, and this is get oxidized by NAD plus coenzyme, and as a result NADH plus H positive is formed. Along with this oxidation, this phosphoglyceraldehyde get phosphorylated also. But now we are not going to add ATP, we are not going to use ATP. This phosphorylation is done by H3PO4. So that will going to provide the inorganic phosphate. So this molecule will converted into 1,3 bis phospho glyceric acid or glycerate. Glyceric acid, because when aldehyde get oxidized, then the acids are formed. So this is glyceraldehyde, it becomes phosphoglyceric acid. Okay, same we have read in the C3 cycle also, that reverse of this. After this, the next step, what is going to this phosphate is going to get removed from here to form ATP. So in our next step, what is going to happen? Synthesis of ATP and conversion of this bisphosphoglyceric acid into 3-phosphoglyceric acid. Okay, so we'll see. By using ADP, ATP is formed. So as I told you, 2 molecules we are going to take, 2 molecules of this, then 2 ADP and 2 ATPs are formed. What will be getting? We'll be getting 3-phosphoglyceric acid. 3-phosphoglyceric acid. Okay, phosphoglyceric acid. Now this phosphoglyceric acid is going to get converted into 2-phosphoglyceric acid or glycerate mutase with the help of the enzyme called mutase. Okay, with the help of the enzyme called mutase. Now, in the next step, what is going to happen? This phosphoglyceric acid is converted into phosphoenol pyruvate. That is by removal of water molecule with the help of enolase enzyme. So this 2-phosphoglycerate, which was isomerized from 3-phosphoglycerate, is now undergo dehydration. That is the removal of water molecule, and as a result of it, what is going to be formed? Phosphoenol pyruvate. Okay, so 2 molecules of this, 2 molecules of this. At this particular position, enolation or dehydration is occurring with the help of enzyme enolase and the removal of water molecule. What we have got? Phosphoenol pyruvate. Now this phosphoenol pyruvate, from here the phosphate is going to get removed to synthesize ATP. So this will convert into, this is going to form pyruvic acid or pyruvate, which is the end product of glycolysis. How many molecules? 2 molecules. 3 carbon containing it is. Okay, and the enzyme which is involved is pyruvate kinase. And ADP is going to take that phosphate, convert it into ATP. Okay, because 2 molecules, so 2 molecules of this also required, and the enzyme is pyruvate kinase. Wherever the phosphorylation is occurring, we are using this kinase enzyme. So this is the end product of glycolysis. 2 molecule of pyruvate or pyruvic acid we have obtained. Now we are going to see how many ATPs we have got and how many we have used. So in the complete process, total ATP used, 2 ATP molecules were used for phosphorylation. 1 was used to phosphorylate glucose and 1 was used to phosphorylate fructose 6-phosphate molecule, and that was converted into fructose 1,6-bisphosphate. So 2 were used, and during this total 2 molecules of ATPs are formed over here and 2 were formed at the time when that step was occurring, synthesis, that phosphate was removed and added in ATP. So it was synthesized when that 1,3-bisphosphate was converted into 3-phosphoglycerate. 2 molecules were formed at that time. So total formed ATP. During the glycolysis are 4 ATP. So out of 4, 2 are used. Net gain of ATP is 2 ATP. It means during the glycolysis 2 ATP molecules are formed, and NADH plus H positive, these are also formed. Two molecules are formed of NADH, and afterward this NADH again converted back to NAD plus to accept more hydrogen and electron. So this is all about glycolysis. Thank you.